All right. So the first couple of talks have talked about kind of, local issues. The stuff I'm going to cover is stuff kind of more at a lake-wide level. And this is stuff that I've been working on for a long time now. But we're also going to be talking about lower trophic levels. So kind of the base of the food web, those things that get anglers really excited. Just I'm going to talk a little bit about my program, talk a little bit about a federal program called CSMI, the Cooperative Science and Monitoring Initiative, and then talk about things that are going on with the benthic community in Lake Michigan, so things on the bottom, and some really exciting zooplankton and larval fish surveys that we did a couple years back. But just a little bit about my research program. I am a research professor at Purdue University. I don't have any teaching responsibilities, so that's kind of nice. Um, I also, with Peter, I work with the Illinois Indiana Sea Grant, and I am their Great Lakes ecosystem specialist. But I'm local. I actually live in Des Plaines. And the reason why I'm local, despite being a Purdue employee, is I work with this program with the EPA called the Great Lakes National Program Office. And so this is the EPA program that's responsible for monitoring the offshore waters of the Great Lakes. And this is their really awesome research vessel. If you guys have ever seen it, it's usually out in the middle of the lake if it's out in the lake, but a lot of times you might see it at docks at Navy Pier sometimes. But it's the biggest research vessel that operates in the Great Lakes. It works in all 5 Great Lakes. And a lot of the data that I'm going to talk about was collected aboard this ship. But one of the things I do with the EPA, with the Great Lakes National Program Office, is I help them with this program called CSMI. And so I just want to make sure that you guys know about this because the CSMI is a really cool program. It's this binational effort that's actually written into the law of the Great Lakes Water Quality Agreement. So in the Great Lakes Water Quality Agreement, there's some language in there that says the parties, the United States and Canada, will put enhanced efforts for science and monitoring into one lake each year. And then that goes on this like rotational cycle. So there's this large scale program that works in one of the lakes each year. So every 5 years it's hitting Lake Michigan, and we're doing enhanced monitoring, enhanced science in Lake Michigan. We don't deal with the Canadians when it's in Lake Michigan, though. We don't, because we don't share a border with them. But in all the rest of the lakes, you have to coordinate with the Canadians. But I just wanted to bring that up because it is a program where there are these groups called the Lake Partnerships, and we actually have a Lake Partnership member here in the room with us today. Vic's part of this group. Where they get together. So it's fisheries managers, but it's also water quality managers, tribal fisheries groups. They all get together and they do other things. But one thing is every 5 years, they need to generate what they call the science priorities. And so basically it's just like, what are the things that you guys need as managers? What kind of science do you need to help you manage the lake? And so then that goes into the CSMI process. So the actual like objectives are driven by management of the lakes, and hopefully they're listening to their constituents. So this is how like the science gets done basically. And as I mentioned, every 5 years we're hitting Lake Michigan. We just happen to be at a place where we can talk about some of the results of the last time that we were in Lake Michigan. So this is kind of the process. So this is just for Lake Michigan. So remember, every year, so this year, the field year is Lake Erie. So pretty soon people are going to start going out. Canadian ships, US research vessels are going to start doing research in Lake Erie based on their science priorities. This is the Lake Michigan CSMI cycle. So the last one, last time priorities were set was 2018. And then there's a year of project planning. There's a year of field sampling that happens. As you can see here, we've got 2020/21. I mean, you guys know what happened in 2020, right? Like COVID wrecked this too. It wrecked everything. So we're a little staggered now, but there was field work that happened, and I'm going to talk about this. That happened in 2020 and in 2021. They couldn't get everything done because of COVID in 1 year. And so then there's a time for data analysis and then a time for reporting out the results. And so that's what I'm doing now, is like kind of talking to groups, letting people know what happened in the last CSMI cycle. Also, so we're actually even in a new cycle now. So I actually know for a fact that probably within the next month or so, the new priorities are going to be released, and those are documents that anybody can access. And so we're actually planning projects for next year, 2025, which will be the next field year on Lake Michigan. So we're sort of doing it again. It's this perpetual cycle. So I just wanted to, so thinking about like from the Lake Partnership's responsibility, they are responsible for identifying the science priorities. I like to ask this question to the group, to different groups. And so just, you can show me on your fingers, like what is the most pressing issue affecting Lake Michigan? If you had this like megaphone where you could talk about what's the issues. So there's, water quality issues in nearshore areas, invasive species, decreased fishing production, or just something else that I haven't thought of. I got a 4. What's the 4?
Invasive species.
Oh, well, okay. So 2. You just doubled up 2. All right. I would agree with that.
And what's it doing to us?
Yeah, I would agree. And that's some of the stuff that's something that comes up all the time with the managers, especially the Quagga mussels. And I'll talk some about that. So this is the last group, the last set of priorities that were set by the CSMI process. So essentially, food web dynamics in a changing ecosystem. This is often changing ecosystem. A lot of times they're talking about Quagga mussels, Dracaenas. Addressing contaminants and bacteria. So again, a lot of times, these are also water quality managers. So they're concerned about, things like beach closures, the effects on tourism, watershed tributary connections and water quality. I thought this was really cool, this aiding and connecting with stakeholders. So part of what the managers want to do is they want to know sort of what you guys think and what your opinions are about how the lake is being run. So these are the priorities that we went with in the last cycle. And then today, what I'm going to talk, so these were the projects that actually were conducted. So they can't address everything, but they can do some focused surveys. One of the things I'm going to talk about is this lake-wide sampling of the broad food web, but especially an emphasis on Dracaenid mussels. There was also a really spatially comprehensive larval fish survey that happened in 2021, and I'll talk about that. A couple of things, though, that this group might be interested. There was a lot of work with lake whitefish, tagging lake whitefish in the way that Luke talked about earlier, acoustic tagging, looking at what lake whitefish are doing in Green Bay, but also on the other side of the lake in northern Lake Michigan. So there's a lot of stuff that was done. I'm not going to be able to cover, a fraction. Right there.
Okay.
So we produced this big document. This is like a 200-page report. We publish it online. And if you guys, that's the QR code to get it. If you guys aren't up for QR codes, you don't want to take a picture, I can make sure that Peter has the link online. But this is all published online, publicly available stuff. All of those different things that I just talked about and the things I'm going to talk about now are covered in detail in this report. Okay. That's part of the process, this reporting out process, is we produce these reports. All right. But now I'm just going to jump into it here. One of the things that happens every CSMI cycle are these big lake-wide assessment surveys, especially focused on benthic community and Dracaena. So this is a huge effort. Again, remember, this is the Lake Guardian, the big EPA research vessel. So they went out and they sampled benthos from 99 stations. So all of the different dots and triangles here are where they're collecting information about the benthic community there. And what they find is that there have been major changes to the benthic community over time. I want you to notice, like, so these happen about every 5 years, right? So this is when these CSMI surveys are when we get this information about the benthic community. So this is looking at the distribution of quaggas throughout the lake through time. When they first started doing this, there weren't quaggas here. There were zebra mussels were the thing then. But quagga mussels soon replaced them. By 2000s, we're starting to see them up in the northern parts of the lake. By 2005, they're pretty much have a whole lake distribution. And then what you can kind of see is through time, they just keep filling into the deeper parts of the lake. But at this point out there in the lake, quagga mussels, 99% of the sites had them. Quagga mussels represent 75% of the benthic invertebrate density and over 99% of the biomass. So almost everything out there that's on the bottom of the lake at this point, from a biomass perspective, is quagga mussels.
They can go to any depth.
They're getting there. So they grow more slowly in the deeper water because it's cold. But I'll show some graphs here in a second. I'll have it broken out by depth. And that's the area where they're actually expanding, but it's slow. Besides quagga mussels, the next most common thing is oligochaetes. So these are small worms. 20% of the density, but again, less than 1% of the biomass.
Just the black there, meaning that was the biggest concentration.
That's the highest density. Yeah, sorry.
Years, it's kind of.
It moves. Yeah, it kind of moves around, and this is all like relative to a given year. So each year it's increasing, but the black's definitely where like you see the most quagga mussels.
It's more prevalent on the top because there's bird. Of Michigan, is that a factor? Or?
I think it's also substrate. They do a little better on. They can colonize sand, but they do a little better on hard substrates. There's more hard substrate up there. So yeah, the benthic communities, they don't differ by like whether you're in the northern part of the lake or the southern part of the lake. It's more about depth. So different communities at different depth. There was a mention of diparia. They'd continue to decline. So we also, during these benthic assessments, they also look at diparia. So the gentleman who talked about diparia was right. This is a very important prey item for fish, especially the native fish in Lake Michigan. At the start of the survey, they're doing pretty well, kind of captured everywhere. But you can see it's kind of the mirror image or the opposite of what's happening with the dreissenids. Now they're only kind of hanging on out in the deepest parts of the lake, and they're not found at very high density at this point. This is the broken down by depth. So this is looking at dreissena. This is like just kind of through time. So the population has increased, but it's kind of leveling off. It's increased by about 13% between 2015 and 2021. And it kind of depends where you are in terms of what's happening here. So we actually saw an uptick in the shallowest areas, but these were really small. These guys might not make it, but we did see an uptick in quagga mussels in the shallow areas, leveled off in mid depths, and it's just a steady increase in the deepest part of the lake. So they're colonizing the deepest part of the lake, but the growth is really slow because it's really cold.
They plankton eaters?
They're filter feeders. They filter out phytoplankton for the most part. Just one thing that did come up that was kind of interesting. There's another non-native mollusk now that's into the lake, the New Zealand mud snail. So there's something new. Don't really know what to think about this one, but we'll see, I guess. All right, so just to summarize, obviously the quagga mussels are dominating the benthic community. They're over 99% of the biomass that's out there. It seems like the population is stabilizing. We know that having all of this biomass of mussels out there filtering the water has kind of done a number on the ecosystem. But you know, maybe we're at a point where like that effect is going to level off. You know, maybe it's not going to get worse. um, Unfortunately, diaphorea continues to decline. This is not just a Lake Michigan thing. We do these same surveys on all the lakes, and diaphorea populations have crashed. They're even decreasing in Lake Superior, which was kind of the last holdout.
What do you think the cause of the decrease in the diaphorea?
I mean, it's
Could it be the
that's the main. I mean, that's the main. If you look at those distributions, it's pretty obvious. So the main thought is competition, but I'm not 100% sold on that. There are a lot of people who know a lot more, think a lot more about this stuff than I do. Like I've heard things that there might be some sort of virus that is like kind of the quagga beds are kind of a great place for the virus to propagate, because diaphorea, apparently this has happened to them in Europe and it was associated with the virus. So I don't know. I mean, it's speculation. But this is, I mentioned they also took video. So that's what the bottom of Lake Michigan looks like out in the deepest parts of the lake. Just, you know, thousands of Dreissenids. All right. I kind of already touched on this. But yeah, so the population of the trajectory of the Dreissenids is kind of leveled off. You know, so hopefully, you know, we're not going to see any big, you know, dips in productivity any further than what they already have done. And that's kind of where I think everybody is with this. All right, so it's a cheery picture, I know. But now I want to talk about larval fish surveys that happen. So we know, you know, the Lake Michigan ecosystem has undergone some pretty huge biological changes, changes in nutrient inputs, invasive species. We've seen a decrease in primary productivity, and it has a similar decrease in zooplankton abundance. And, you know, we've seen a lot of kind of lake-wide low fish recruitment events in the last, you know, several decades. One thing that we're learning from doing these big lake-wide assessments, though, is like not all these the changes aren't just evenly spread out throughout the lake. So it kind of depends on where you are in terms of the effects, the ecosystem effects that are being seen. One of the things we did, a former student of mine did, was we went back and we looked at every day. There's a satellite that flies over the Great Lakes and takes a picture. So we worked with some people who were like optical physicists, and they figured out that you could actually look at the pixels in those pictures and look at the ratios of the different colors and relate that to chlorophyll. So the amount of primary production. So we had, and these satellites have been flying over since the 80s. So we went back and mined this and like looked at, can we see changes in productivity based on these satellite images? And so this is the, like kind of the take home here. This is Lake Michigan. This is an average from the month of May, July, and September. And this is from 2000 through about 2018. And then I have this here. These different colors represent the different depths of the lake. So red is shallow out to blue. And if you can see it, these lines are color coded too. So these are all these trends by depth. So we did all this work, which you guys probably already know this, right? We're not seeing a decrease in primary productivity in shallow areas. So these red lines are straight across. Where we see the dips are in the blues and the greens. So the productivity losses that we're seeing in the lakes, they're not in nearshore areas. In some places, we're seeing increases. The lines are going up. But pretty much consistently in the blues and the greens, we're seeing decreases. So offshore primary productivity is decreasing. And you guys see that with the clear water, right? It's much clearer now. So we went to a lot of effort to do that. And so just to say, and the other thing is that I don't have the results here, but we split the lake out into 4 different quadrants because you see different patterns in different parts of the lakes. These offshore decreases are more pronounced in the southern basin, the southern part of the lake. we also have a student who's looking at some models to model fish recruitment. So he's taking the conditions. We have these really good descriptions of the conditions of the lake. We can plug that into a model. So we kind of have like these virtual fish that are experiencing these different conditions, and we see if they survive or not. And we see if they survive in different parts of the lake. And so you can think about this as like, according to what we know about fish recruitment, which we don't know everything, a model's only as good as what your assumptions are and the data that you put in. But if what we know about yellow perch is correct, most of the fish in Lake Michigan are coming from Green Bay. That's not necessarily that surprising. Green Bay by far is the most productive area. But this is northeast, northwest, northeast, southwest, southeast. But in the main part of the basin, we should be seeing more recruitment for yellow perch coming from the southern basin of the lake and not the northern basin. This is based on a lot of things: temperature, food availability. Those are different years. So we actually, so these colors these were years where they actually had high year classes of yellow perch. And so our model accurately predicts that, like, when you have a good year, our model projects good years. The blues are bad years and the grays are in between. So these are based on like historical data. And then anyway, the take home here is that different parts of the lake produce different amounts of fish. And we ran this for yellow perch. We ran it for alewife too. And it's a similar thing. So according to our models, we should be getting the majority of the production of the fish for alewife and for yellow perch in the main basin in the main lake should be coming out of the southern basin. So I say that because we don't really have a lot of data, to go with this. These are virtual fish swimming around in a virtual lake. But that's why we designed these larval fish surveys, because we actually wanted to see if there's differences in production of at least larval fish in different parts of the lake. So again, I say, like, one of the reasons why I brought up that whole CSMI thing is because we care about alewife now, because anglers they're very adamant about, they want the salmon to have food. So it's made its way all the way up to this. So we're actually doing larval fish surveys looking for alewife, which as an ecologist, I never would have thought I was doing this, but here I am. Let's see. So the reason why we care about alewife is they are the most common prey for salmon in the lake, salmonids in the lake. Their biomass is pretty low right now, so that's why people are concerned. And they've been persistently low for a while. So we want to know kind of like what is behind these trends. So this is the survey that we conducted in 2021. So there are 15 different transects here. And at each transect, there are 7 stations going out from nearshore to offshore. This is a huge effort. This was all done during the month of July because that's when we think alewife are hatching. This was done by 3 different federal ships, all working at the same time. So the top is the RV Lake Explorer, which is out of Duluth, Minnesota. The middle one is the Lake Guardian. The bottom is the Sturgeon, which is a USGS vessel based out of Sheboygan. So all of this work is happening at the same time during the month of July. And they're all going out trying to look at looking for larval fish, primarily alewife, but also looking at just general conditions and zooplankton abundance, which I'm also going to talk about because that's the food. Another thing that people are concerned about is UV light. So we all know that the lake is much clearer. We've established that. Alewife actually are very sensitive to UV light. And so the idea is that maybe that increased water clarity is something that's like killing the alewife. Maybe the alewife want to hang around near the surface. Maybe they're getting zapped by UV. So we did all of our sampling was done at 3 different depths. So at the surface, at the 1% UV, and then when there's no UV below that. Okay, so that's something that'll come out in the results. The other thing that we're concerned about is the food availability and the food quality. So veligers are the little baby quagga mussels. We know at times they are insanely abundant out in the water, but we don't know. We don't know what eats them. We don't know if there's a nutritional quality to them. We don't know. So we also wanted to go out and look at the food that's available for these fish during the month of July, because we know, well, I said we don't know, but we're starting to find out. We've done some experiments where the fish are fed only veligers and fish are fed a normal zooplankton diet. And so these larval fish had hatched on the same day. And this one was fed veligers and this one was fed zooplankton. So those differences, we're talking about tiny millimeter differences, but that's huge to these fish. And it infers a huge size advantage the faster that you can grow. So we wanted to get an idea of the food that was out there while these alewife are hatching. All right, so get into some of the results. So this is sort of the lake-wide snapshot of what food availability looks like for fish during the month of July. So this is copepod, like small copepods, so small zooplankton. This one is at the UV layer, so like kind of just below the surface, and this is below the UV layer. The size of the circles represents density, and so do the colors. So basically, up here in the northern part of the lake, low zooplankton. In the UV layer, it's also lower. They're more in the southern part of the lake, but there's fewer fish in the UV layer than below that. So you get that the zooplankton are mostly in the southern basin, and they're mostly deeper in the water column. I should also mention all these samples were collected during the day, so we don't know what they're doing at night. But that's the takeaway from, that's where, so this is primarily the food that the larval fish are eating. We were worried about the veligers. This is veliger distribution. We didn't actually catch many. So in the UV layer, there's hardly any veligers, so up near the surface. We did see some pretty high densities of veligers, but it seems like this is kind of like just a quick process. They release a bunch of them and then they go wherever they go. But that doesn't seem to be as big an issue, at least based on what we've seen. There aren't just tons of veligers everywhere, which is what we were kind of afraid of. All right, so now the actual catches of fish. I need to set this up a little bit. So here, these are alewife catches, larval alewife catches. The size of the circle represents the relative number of fish that were caught. This is summarized across all of those transects. So just one per site. And then these different colors represent where the fish were collected, like in what proportion. So yellow is at the surface, green is the 1% UV layer, blue is the deeper water layer. So what you can see is, we see a lot of kind of a, I don't know, strange distribution of where the fish are. We're getting big catches close to here, big catches at Sturgeon Bay. For the most part, I would say that the fish alewife are deeper in the water column. So they're at the 1% UV or no UV layer. We see a surprising lack of fish here. This is kind of where we expected to catch a lot of them. But we also have at Purdue, we had another project where a guy was going out and collecting fish every week. This is just, again, one snapshot. And off of Muskegon, which is, right around in here, they actually caught a ton of larval alewife in June. So we might have missed them. They might have been earlier in this part of the lake. This part of the lake warms up faster, so maybe they had already hatched. We already kind of missed them. Thankfully, though, we do have those Muskegon samples, so we can kind of compare that. But this is what the distribution of alewife larvae looked like in 2021, and by far the biggest effort anybody's ever done to try to look at where alewife larvae are.
You go to the same places at the same time every year?
No, this is a one of 5 year snapshot. This is like, we have done larval fish survey. I'll show some comparisons from year to year. We've never done this like big lake-wide assessment before. We've tried it where we do like 3 sites and we hit them like once a month. We've tried 2 sites and we hit them every week. Like there's been a lot of different effort to do this. We're still trying to figure out kind of what the best, obviously we missed something here. So, all this effort is still not perfect. So this was the target, ALY for the target. We also just happened, just out of pure luck, to be out there at a time when there was a huge bloater hatch in 2021. So bloaters, if you're not like chubs, a native species, and they were everywhere in 2021. And so we were out there dragging nets around. We just happened to catch them. Bloater were our second. So hold on. So alewife were our most prominent catch. So 64% of the larval fish that we caught were alewife. But still, we happened to catch, 24% of our catch was bloater. And they were almost everywhere, like everywhere we looked. A lot of them were offshore. And look at the color here. The bloater tend to be at the surface. And so we don't really know what to do with that, but it's just kind of interesting that they seem to be in different parts of the water column. Bloater, there's no indication that bloater have a problem with UV though. Bloater have their larvae, they have a lot of pigmentation, and so they're actually more resistant to UV than alewife are. So, yeah, and we do know that based on like subsequent surveys, this is what happened. These were the fish that were hatched in 2021. These fish have survived to where there's a decent number of bloater out there now, at least from that 2021 year class. And what's cool is that happened in a lot of lakes. In Lake Superior, there were a ton of bloater and cisco and kai, all these same types of species. All these native planktivores did well in 2021.
Did you find bigger zooplankton up near the surface? That these guys might have been shooting at?
I'd have to look at it. The bigger zooplankton though tend to be, yeah, offshore and deep. Yeah. So, but again, we don't know what they're doing at night. We do know that the big calanoid copepods come up near the surface at night. So maybe. What I also have a student who's at Purdue right now who's looking at the growth of these alewives and the diets. So I don't have that data yet, but we're working on it. All right, so this is just kind of what I was talking about. Like so alewife are avoiding the surface. Maybe they're avoiding it. Maybe they're just dying. Maybe the ones that made it up there just died. Bloater, opposite. They love it at the surface. All right, so this is where we can kind of compare. We did some other larval fish surveys in the 2025 or 2015 CSMI survey. Here, this is kind of typical of what we've seen over the last several decades where at least during the summer months, you tend to catch mostly alewife. That bloater catch in 2021 was kind of a jackpot kind of thing. Alewife density from 2021 is actually lower than 2015, but these results are, again, 2015 was just a few stations. It wasn't the big lake-wide thing. So I'm not sure about how much that really means. But bloater density went up in the 2021 based on 2015. All right. So in terms of just the key findings, the zooplankton community is kind of the same as it was. It's indicative of a cold lake that doesn't have a lot of productivity. Calanoid copepods are the dominant zooplankton out there. We didn't see a lot of veligers. That doesn't mean that there aren't a lot of veligers at certain times. It means that our sampling design probably just didn't do a good job. We know that there's a lot of veligers because there's a heck of a lot of mussels out there. When we were doing these surveys, we were seeing zooplankton densities that were higher in kind of the deeper water that we sampled. We also saw the alewife were avoiding the surface. And we think at least, and we can probably ask some of the people in this room probably know better than me. But I know that the bloater recruitment seemed to do pretty well for the 2021 year class. And the thought is that the larval catches that we had of alewife are relatively comparable to what we've seen in the past. Okay, in terms of the management implications of this, it's still kind of to be determined about UV being a sort of a source of mortality for alewife. We certainly didn't catch them in the high UV areas. This could mean that like different habitats are potentially good sources for alewife. Green Bay doesn't have the same UV problem that the open lake does. So maybe that's where the alewife recruits are coming from. Let's see. Yeah. The growth and diet of alewife may be similar to the low values that we've seen. So we do tend to see slow growth for alewife in terms of like where they are now versus where they were from previous studies. Again, I have a student who's working on this now. I said that it's really hard to compare the catches of alewife with this survey compared to previous ones, but one thing that we can look at their growth rates and compare that, and we can see if the growth of larval alewife is continuing. It's been slowing through time, which is a bad thing for alewife survival. But we'll see what happens with 2021 here very soon. So I want to be very clear that I didn't even really do a fraction of this work. This is the hard work of a lot of people. I helped coordinate it. I helped get the word out. But there are a lot of people who worked really hard on all this stuff and deserve a lot of credit. And if you're interested in what the findings were, again, that's the QR code for the report. So thanks for your attention.
We have time for questions. If anybody has any questions for Paris?
And your feeder fish, are they improving? Like the gizzard shad or some of them?
So the work that we're doing, you saw the size of those boats, right? Like everything we're doing is pretty much offshore. That's one of the critiques of the CSMI is that's the equipment that we have. So we use big boats and we're doing things that are like really far offshore for the most part. So we're thinking more about like, populations of lives and stuff like that. So we don't really do much, gizzard shad is much more near shore distribution than alewife. So I can't really say much about
Where
what they're
is it shed with deep water or middle?
shallow, very shallow. Yeah.
Okay.
Yeah.
Any thought been given to change the timing of the sampling? Like you guys usually do July, like they talked about, and it's a significant amount of fish June. Have they thought and given like doing sampling at other time?
Yeah. So the question was, so if we thought about sampling at different times, and we definitely did. We definitely do. If you care about something other than alewife, we're not really giving you information about that. Like if you care about lake whitefish, we're not there at the same time. So yeah, we've thought about it. We would also like to move way closer to shore. We think we're missing a lot of things that are near shore. So there's definitely gaps in what we're doing. But the thing is we've done this now. We've done these bigger surveys probably like 3 or 4 times. And so there's a real a need and a desire to sort of let's put our heads together and let's think about a way to do this, that makes sense, that we can get the best information. I still think we're probably going to target alewife.
Well, yeah, again, that's
Yeah.
particularly.
Yeah. So it's kind of interesting that, and it's a nice coincidence that there is the grad student at Purdue who's doing the alewife work, because he worked his butt off, like for 2 years, went out every week and was sampling larval fish every week. And so it will be really interesting to look at his stuff and then compare it to ours and see like what kind of information we missed, you know, versus he's only sampling in like one spot. Yeah, yeah. Yeah. But that's what we need to do. We need to like try to figure out how to do the best that we can do with the resources that we have.
Something like that would be incorporated at least. Go against your team.
Yeah, I didn't even mention, I just kind of skipped it over it during the slide, but it was actually incorporated into the survey, but it's just not on here. Or no, it is. So Purdue, that guy. So that's where this guy was sampling every week. So we can compare what we found at Grand Haven to what he did.
I'm wondering, we see now when we catch lake trout and brown trout on the charters, most of what they're eating are gobies. And I'm wondering what that does to the overall alewife population. I mean, assuming they started eating the gobies because alewives aren't available, but are the alewives gonna come back and they still eat the gobies? Is that gonna affect overall alewife population?
Yeah, so there also with CSMI, there's a guy at Michigan State who's been looking like long-term at salmonid diets, you know, he's probably got, I don't know, 7 or 8 years of data now. And so what we know is that, you know, like, yeah, lake trout, they have a pretty diverse diet. We don't get a lot of browns in that. They don't get a lot of browns in that, but I think that would be a pretty diverse diet. You know, steelhead, relatively diverse, but the Chinook are eating pretty much just alewife.
True, but coho is.
Yeah.
Staging now, we're catching king salmon with gobies in it.
Cool, that's great.
And
I know that like the coho, the story is they go real far down south and they eat mysids. They eat the little invertebrates.
Little shrimp,
Yeah, yep.
That, for us here, that's deep water. Salmon fishing, and then, when they're in close to shore, it's changed the way that we
yeah, they
fish.
about half of them.
Or,
We fish lower toward the bottom now a lot of times, and you can tell half the coho or half the alewife.
Yeah.
So,
That's good. There's there's not a shortage of gobies out there.
no.
So the problem with gobies though is that they're really hard to sample. It's really hard to get an
Sure.
idea how many are out
Well,
there.
yeah.
There's a CSMI process that's been ongoing. Vic, did you did you have a comment? Yeah, exactly. Yeah, so there's a group, it started as a CSMI project, but now he's got long-term funding to where they're looking at like imaging, like they're using like remote underwater vehicles to like take pictures and count the gobies.
Automated learning to like pick gobies out of the pictures and measure them.
Yeah. So they've been working on that since 2017.